I'd like to introduce Myia St John. Myia is a PhD candidate, a speech pathologist and research assistant in speech and language. She's currently based at the Murdoch Children's Research Institute and University of Melbourne. And before completing her Masters of Speech Pathology in 2006, Myia worked for a number of years in specialist school settings. So working with children with intellectual disability and I'm guessing speech and language conditions as well. The work fostered her interest and passion for working with paediatric intellectual disability and rare diseases. Myia has since researched a number of rare diseases such as Klinefelter syndrome, NF type 1, and focuses on characterising the communication profiles in these conditions. She is currently completing her PhD investigating speech and language across a number of rare conditions, including Cat6A and Coolen. And I was pleased to read when I had a quick look through at Myia's presentation beforehand, she also has an interest in my daughter's condition, FOXP1 syndrome. So I will pass you over to Myia and let her take her presentation away. Thanks, Myia.
Thank you everyone for coming along to the event today and for supporting Rare Disease Day. My name is Myia St John and it's a privilege to speak about my work and to share my experience as a researcher, a speech pathologist and as a PhD candidate. I'm based at the Murdoch Children's Research Institute within the Centre of Research Excellence in Speech and Language. And this is an international collaboration of experts in the fields of speech pathology, paediatric neurology, neuroscience, genetics and bioinformatics. And the core vision of this research group is to transform speech pathology practice by identifying and targeting the underlying causes of developmental speech and language disorders. So we know that speech and language disorders arise from genetic, neurological and environmental factors. And we know that genetic factors are thought to play a particularly significant role in speech and language development. One of the key goals of the speech and language CRE is to recruit children who have complex speech disorders, such as apraxia of speech, and to then perform genetic sequencing in order to identify the genes that may be responsible for their speech challenges. And once a potential gene has been identified as potentially impacting speech, we can then attempt to find a group of others who have this gene change and then see if that gene change causes a similar profile of speech and language across the entire group. As part of my current PhD project, I'm investigating a number of these genetic conditions in order to unravel their speech and language profile. One of these conditions is Cat6A syndrome, which is the reason I came to be a part of this event today via a few of the amazing advocate mums who work and really embody the Cat6A Foundation. And as an example of the work that I do, I'll focus a little bit on Cat6A to show what the process looks like when we investigate any given condition. Like many rare conditions, Cat6A syndrome is still being understood. And there's only roughly 300 people diagnosed worldwide, or I think actually maybe 350. And it occurs when there is a variation, like a spelling mistake to the Cat6A gene, meaning that the gene loses or alters its usual function. And because the Cat6A protein is involved in the expression of many other genes, a variation in the Cat6A gene can cause changes in many different parts of the body. And this isn't unique to the Cat6A gene. Many gene syndromes can cause difficulties or issues or complexities in different parts of the body. So you may have issues with heart or brain or speech and language. Known features of Cat6A include intellectual disability, low muscle tone and distinct facial features. And research has also described speech delay using terms such as strikingly delayed or absent speech. Oftentimes, terms such as speech delay are used by medical professionals, and although it can appear or might appear sufficient to just say, well, those with Cat6A have significant speech delay or significant communication delay, it doesn't really provide enough nuance to understand the best treatment options and strategies required. And so very simplistically, we can divide communication into two distinct areas: speech and language. Speech being the perception and production of speech sounds, while language is the production and understanding of words and grammar to communicate meaning. So put simply, if a child can't say or doesn't say a particular word, is it because they can't produce the sounds or string of sounds in that word, which would be a speech problem? Or is it because they don't have the concept of that word in their vocabulary? They don't know what that word means, and that would be an issue with language. We can also further break down each of these areas into distinct disorders. So under the category of language, you could have issues with receptive language, which is what you understand, or expressive language, what you can say, or you might have issues with social language. You could also have a number of different speech disorders, and each of these are distinct in how they present, but also how common they are, how we would treat them, and how easily we can treat them. So phonological issues and articulation disorders, for example, and for the most part are more common and they're more easily treatable, whereas childhood apraxia of speech, or CAS as we call it, is very rare and it's more difficult to treat. And so knowing these distinctions in speech disorders or language disorders is super important, because the treatments and the strategies and the approaches that we would use for each of these conditions will be different, and unpacking these for individual genetic conditions is our focus. And so in our Cat6A study, for example, we've recruited 49 individuals from all over the world with pathogenic Cat6A variants, and we've assessed each person using a series of questionnaires and assessments. And we tailor these questionnaires and assessments according to their abilities and their age. So we had individuals from 1 year up to 31 years. We first looked at their medical and neurodevelopmental profile and found that in Cat6A, many have intellectual disability, many have vision problems, many have issues with gastrointestinal concerns, sleep disturbances, and so on. And even though we're focusing on speech and language, it's always important to look at the speech and language within the profile within the entire medical and neurodevelopmental profile, because as you can imagine, some of these things will impact a child or an adult's ability to communicate. Vision problems in 74% of this group, for example, may impact their ability to read. Hearing loss, for example, has a large impact on speech development. So we always have to think about these other features alongside the communication, the speech and the language. We looked also more specifically at feeding, and we found that individuals clearly had common and persistent feeding difficulties in Cat6A, with over 90% affected at some point in their life, and many still had feeding challenges beyond their teen years. So this was an important thing to consider in the Cat6A group. Not surprisingly, considering the links between feeding development and speech development, and considering there were persistent feeding issues, communication impairments were also striking, and we found that actually 73% of this group were either non-verbal or minimally verbal, often into late childhood and beyond, so into the adolescent and adulthood. So I'm going to show a video, and this video depicts a young boy with Cat6A. He's currently minimally verbal, but he does display some basic receptive language skills, so his understanding of language. He understands some simple vocabulary and some simple following of directions. And this was a common presentation of what Cat6A presented as in this group.
Think. Where's the horse?
Look for the horse.
Horse. Good listening. Where's the sheep? Sheep. Good.
So although 73% of the group did not use verbal communication, we were able to utilise an assessment tool that captured informal communication behaviours for those who couldn't speak. And we found that in Cat 6A, most individuals used alternative methods to communicate wants and needs. So when examining social behaviours like greeting others, requesting needs, making choices, and communicating feelings, those with Cat 6A relied on behaviours like body and limb movements, non-verbal vocalisations like grunts or cries, and face and eye movements to communicate. Sign language and picture communication devices were also used. However, this was often complicated by the child's motor abilities, as well as their access to devices and their access to therapists who were able to facilitate the use of the devices. Across those who were verbal, which were 13 in our group, communication milestones were often delayed, and most individuals had complex and co-occurring speech diagnoses even when they developed speech. And so the diagram shows that a number of distinct speech disorders, and you can see that most individuals were placed at a point of intersection. So phonological delay, as an example, was most commonly diagnosed, but never on its own, and always within the context of another condition. So it was always co-occurring with another speech diagnosis, which made children more difficult to understand. In summary, for Cat 6A, we can see that for many individuals, traditional verbal communication remained a challenge into adolescence and adulthood. And so it's important for these individuals with Cat 6A for both traditional motor speech therapies as well as alternative and augmentative communication options like picture communication devices or speech generating devices should be implemented as early as possible to ensure the best communication outcomes and to reduce frustration for both the child and the parent as well. But amongst the other conditions our group has studied, so we've studied a number of genes, this profile of many non-verbal communicators isn't always the case. Each syndrome is unique, and it's why we look at each of these conditions individually. And some of these conditions show a very different profile of communication. Cool and de Vries syndrome, as one example, was characterized by low muscle tone and oral apraxia, which suggests intensive motor speech therapy in the early years to facilitate the development of first words. Other conditions like Klinefelter syndrome, which is something else I've looked at, were a very mixed group. So while some individuals developed very clear speech without any speech therapy required, others presented with more complex issues with social communication or language and literacy difficulties later in childhood. And so these nuances between conditions emphasize that the understanding of the underlying genetic cause is so important for treatment planning, and ultimately each child is unique regardless of their rare condition, but it's most important to see which factors, genetic or otherwise, will impact their trajectory and their quality of life. And in summary, I'd just like to thank all the participants and families who have been involved in any of our research studies across the speech and language CRE. I'd like to thank Meg Salisbury and Jane Alul, associated with the Cat 6A Foundation, Heather Renton from SWAN, Deborah Cameron from Wellways, both of which were part of inviting me to come and do this talk, my supervisors, my PhD supervisors, Professor Angela Morgan and Professor David Amor, and of course the entire Speech and Language Centre of Research Excellence team. Thank you.